Hello friends. Today we are talking about the ligand protein ligand interactions that can be described quantitatively. So how can we describe it quantitatively? So firstly we will take a basic equation. Here P stands for protein and L stands for ligand. So the equilibrium expression for that is P plus L give rise to PL, that is in bound form. So the equilibrium constant Ka that can be expressed as PL, the molar concentration of PL upon molar concentration of P into molar concentration of L. So after rearranging this equation, we can have KAL. We can take this L here. So Ka into molar concentration of L is equivalent to ratio of bound protein to free protein. So we can see that the free ligand is directly proportional to this ratio. So if we suppose that the concentration of free ligand is very high as compared to the free binding sites. So if the concentration of free ligand is very high, so the concentration of ligand free ligand remain constant. Next, we will talk about theta. What is theta? Theta is the fraction of ligand binding sites that have been occupied. So, how we can express this? That is binding site that are occupied upon total binding sites. So, the occupied sites are PL, and the total binding sites, that will be the free binding sites and the occupied binding sites. So this expression we will get here, and we can substitute the value of PL from here, from equation 1. The value of PL can be substituted as Ka into molar concentration of L into molar concentration of P. So we will substitute this value here, and after substituting we will get theta is equal to L upon L plus 1 upon Ka. So Ka, we know that K is the association constant, that is equal to Kd, that is dissociation constant. So obviously if Ka is more, that means the affinity of a ligand towards one particular protein is high. Okay, here we can see this thing. If Ka is high, then affinity of a ligand towards protein is also high, and it can be inverse in case of Kd, that is dissociation constant. Kd is high, then affinity between a ligand and protein will less. So we can substitute the value of k 1 upon Ka, that is equivalent to Kd. So this value can be substituted, and the final expression for theta is this. And if we draw a curve between theta, that is the fraction of ligand binding sites occupied, that is plotted against the concentration of free ligand, then we will get a hyperbola. So hyperbola we are getting here, and here we will get Kd, value of Kd, the value of L at which the half of the available ligand binding sites, this is 0.5, the half of the available ligand binding sites are occupied, that is the point Kd. Okay, on x-axis. I'll repeat one more time. The L at which half of the available ligand binding sites are occupied, that will equivalent to Kd. So so finally we can say, if the affinity of a protein and a ligand is high, so the value of kd will be less. Next, we are talking about the cooperative ligand binding of hemoglobin that can be described quantitatively. Here we have protein, and n represents the number of binding sites. So n is for number of binding sites. So normal equation P plus NL, that will give rise to the bound form, the protein and ligand that are found in bound form. So the association constant, that will become PLN, molar concentration of PLN upon molar concentration of protein into molar concentration of free ligand with n binding sites. So theta we have already discussed. Theta is the binding sites occupied upon total binding sites. So from that expression, we can see here, we can get the expression that is L n upon Ln plus Kd. After rearranging this equation, we will get theta upon 1 minus theta is equal to molar concentration of L raised to power n upon Kd. So taking log on both sides, we will get log theta upon 1 minus theta is equal to n log L plus log minus log Kd. So the n here, that is NH, we can represent it as NH. Why? Because this is Hill equation. This equation is known as Hill equation. So the n here is Hill coefficient, NH, which is basically a slope. Now we will talk about NH, that is Hill coefficient, that is NH. We will discuss about its values. If NH is equal to 1, what does that mean? That means ligand binding is not cooperative. Not cooperative. This can be in a multi-subunit protein where the subunits do not communicate at all. So in that case, NH will be equal to 1. If NH is greater than 1, that means positive cooperative. Okay, positive cooperative means a situation just like in hemoglobin, in which the binding would be completely cooperative. All binding sites on the protein will bind to the ligand simultaneously. But the thing is, if we talk about hemoglobin, there is n is equal to 4. So so NH can't be equal to n. So NH for hemoglobin, NH can't be equivalent to 4, because theoretically we can say that the all 4 subunits of hemoglobin get attached to the ligand or to the oxygen, but practically it is not possible. So NH is not equivalent to n, but if it is greater than 1, like for hemoglobin, NH is equal to 3 can be seen, but not equal to 4. So if NH is greater than 1, then positive cooperativity can be seen. And the third case, if NH is less than 1, that means negative cooperativity. What does that mean? That means the binding of one molecule of ligand impedes the binding of others. So that is the negative cooperativity. Thanks for watching.